Module 1, DNA cloning and PCR. Defining the genetic basis of disease requires the analysis of DNA and sometimes chromosomes. The vast majority of our genetic material is organized as immensely long DNA molecules, and changing just a single nucleotide out of the more than 3,000 million nucleotides in our genome may cause disease. Sophisticated technologies have been developed to purify and manipulate genes, enabling studies on how they work and how they can be used therapeutically, and providing ways of analyzing mutations and studying the molecular basis of disease. In Chapter 3, we describe the core technologies for purifying and analyzing DNA sequences. DNA cloning, fractionating and purifying DNA by transforming cells with recombinant DNA. Cloning DNA in cells is a way of purifying DNA sequences. Large amounts of many identical copies of a DNA sequence can be prepared so that the sequence can be studied or put to some use. This slide shows transformation as a way of fractionating a complex sample of DNA fragments. The key point is that transformation is selective. When a cell is transformed, it usually picks up a single DNA molecule from the environment, and so different fragments are taken up by different cells. For simplicity, this figure shows only the DNA sequences that are to be cloned. In practice, they would be joined to a vector molecule to make a recombinant DNA that is typically circular. Cell clones can be formed by cell division from a single transformed cell and be propagated to produce a large number of cells with an identical foreign DNA sequence that can then be purified after the cells have been broken open. In the transformation procedure, a complex mixture of DNA molecules in solution is added to a suitable cell population. The process of transformation is the mixing of the DNA with the cells. Transformed cells typically uptake only one foreign DNA sequence. These cells can then be selected and amplified from a single transformed cell. Very large number of cell clones, each with the same foreign DNA sequence, descends from a single transformed cell. Following amplification of cell growth, cells can be lysed and the DNA can be purified, obtaining many identical copies of a DNA sequence or clone. DNA cloning in bacterial cells, DNA copy number amplification and separation of clones from different transformed cells. For a cell transformed by a recombinant DNA molecule, an increase in the cell number leads to a proportional expansion in copy number of that recombinant DNA. Growth occurs originally in a solid medium after the transformed cells have been spread out on a plate of agar containing a selective antibiotic. Individual cells will be physically dispersed to different parts of the plate, and individual surviving cells, those that harbor the recombinant plasmid with the selectable marker, then go through several rounds of cell division in situ to form separate viable colonies. An individual colony can be picked or removed and allowed to go through a second round of amplification by growth in a liquid culture medium. For simplicity, the cloned DNA fragments are shown in the absence of the vector molecule. In panel B below, vectors have their own replication origin and can replicate within a bacterial cell independently of the host chromosome, often replicating much more frequently than the host cell chromosome. For simplicity, the illustration here shows a very modest threefold amplification of the recombinant DNA, but some plasmids allow amplification to 100 copies or more in bacterial cells, leading to a large increase in copy number of the DNA clones. Of cloning vectors. Several types of cloning vectors are available for use in the laboratory. These include the plasmid, the bacterial artificial chromosome, otherwise known as BAC, and the yeast artificial chromosome, or YAC. Their host cell, maximum cloning capacity, and utilizations all differ. First, let's consider the plasmid. The plasmid is the workhorse of the recombinant DNA laboratory, and its host cell is E. coli. In fact, plasmids were first identified as naturally occurring extrachromosomal circular pieces of DNA that aid in the transmission of bacterial resistance in nature. These have been exploited for use in the laboratory and modified to a great extent. They have maximum cloning capacities of anywhere from 5 to 10 kb, depending on which type of plasmid is being worked with. Plasmids are sometimes used, or the name plasmid is sometimes used interchangeably with the word vector. So plasmid and vector often are referred to in the same context. Plasmids replicate independent of their host chromosome. They have their own origin of replication and can reach high copy number in the sense that multiple copy numbers of the plasmid occur within the cell. These are widely used for cloning small DNA fragments, and in addition to an origin of replication, plasmids typically contain some type of selectable marker, that is a way that scientists can tell whether or not that plasmid is in the E. coli cell. Typically, that selectable marker is a genetic element that conveys antibiotic resistance. So, for example, if a E. coli cell were transformed with a plasmid that had an ampicillin resistance marker, then those cells would be able to grow in the presence of ampicillin, and thus one would know that that cell contained that particular plasmid. The bacterial artificial chromosome, or BAC, again, is an E. coli host cell cloning vector. It has a very large maximum cloning capacity, and it has a very tight constraint on copy number that allows very large fragments to be stably propagated. Bacterial artificial chromosomes, or BACs, were very useful in sequencing genomes when we needed to create libraries of very large copies of DNA. The yeast artificial chromosome is similar to that of the BAC in that it can contain a very large segment of DNA. It has a maximum cloning capacity of greater than 1 megabase. Essentially, the DNA that is carried in the YAC are small linear chromosomes that consist primarily of human or other foreign DNA. So it mimics the chromosome of the yeast, and they're linear, so that is different than the BAC or the plasmid. And again, these were very useful for creating large libraries that could be then propagated in the yeast Saccharomyces cerevisiae. Of DNA by restriction endonucleases. Restriction endonucleases, otherwise known as restriction enzymes, are the molecular scissors which are an integral part of the creation of recombinant DNA or cloning. Restriction endonucleases cut at specific sequences and may cut in a symmetric or asymmetric fashion. Shown here is the cleavage pattern of the restriction endonuclease EcoR1. EcoR1 cuts at the sequence GAATTC. In other words, whenever the restriction endonuclease EcoR1 encounters the sequence GAATTC, it will cleave the double-stranded DNA in a specific fashion. As you can see, the restriction endonuclease EcoR1 cleaves asymmetrically, that is, that it cuts in a staggered sort of fashion, as exemplified by this slide. Each restriction endonuclease has a unique sequence, and it is only at that sequence that the enzyme will cut. Restriction endonucleases are naturally occurring enzymes that occur in bacteria and act as a primitive immune system to cleave foreign DNA on bacteriophages that infect the cell. Restriction endonucleases have been exploited for use in the laboratory, and now these are available in recombinant form. They are highly invaluable and make DNA cloning possible. Recombinant DNA using DNA ligase. In order to complete the cloning process or the creation of recombinant DNA, one needs to ligate together the two fragments that are generated. One needs to combine the vector or the plasmid with the insert DNA or DNA fragment. This DNA fragment could be obtained through isolation using restriction endonucleases. Alternatively, it could be made available through polymerase chain reaction. At any rate, there is a particular sequence within the vector known as the restriction endonuclease cloning site or the multiple cloning site. And this particular region of the vector will have the sequences of many different restriction endonuclease. For simplicity, the single ECOR1 site is shown in this site, but typically a vector would contain multiple restriction sites. When one combines in vitro the DNA fragment with the cut vector and adds the ligase enzyme will then form a phosphodiester bond between the DNA fragment and the cut vector. Because the two ends of the vector and the DNA fragment have been cut with the same restriction endonuclease, and this is a necessary prerequisite, these ends then will base pair with one another because they're homologous in sequence. Then, although they're hydrogen bonded, they do need the ligase to come and form the phosphodiester bond so that this creates a permanent covalently linked molecule. Polymerase chain reaction. Polymerase chain reaction is a phenomenal laboratory technique that revolutionized the world of biotechnology and recombinant DNA, and it has implications in every facet of laboratory practice. The polymerase chain reaction usually consists of 25 to 30 cycles of denaturation, annealing of oligonucleotide primers that flank the desired sequence, followed by new DNA synthesis of the desired sequence. This figure shows the fundamental steps of the polymerase chain reaction. Numbers in the vertical strips on the left indicate the origin of the DNA strands, with original DNA strands represented by 0 and PCR products by 1, made in the first cycle, 2, a strand made in the second cycle, or 3, a strand made in the third cycle. The first cycle will result in new types of DNA product with a fixed 5' end, which is determined by the primer, and a variable 3' end extending past the other primer. After the second cycle, there will be 2 more products with variable 3' ends, but also 2 desired products of fixed length, shown at the left by the filled red squares, with both 5' and 3' ends defined by the primer sequences. Whereas the products with variable 3' ends increase arithmetically, the amount is equal to 2n, where n is the number of cycles, the desired amplified products initially increase exponentially until the reaction reaches a stationary phase as the amount of reactants become depleted. After 25 cycles or so, the desired product accounts for the vast majority of DNA strands. PCR essentially is DNA synthesis in a test tube and requires primers, deoxynucleotides, and a thermostable DNA polymerase. This technique was so enormous to the scientific community
that Kary Mullis, who's invented this technique, received a Nobel Prize for his invention. This slide depicts the different phases in a PCR reaction. After a lag phase, the amount of PCR product increases gradually at first in the exponential phase, beginning after about 16 to 18 cycles, and continuing to approximately the 25th cycle. The amount of PCR product is taken to be proportional to the amount of input DNA. Quantitative PCR measurements are made on this basis. With further cycles, the amount of product increases at first, but then tails off as the saturation phase approaches. When the reaction efficiently diminishes, as reaction products increasingly compete with the remaining primer molecules for template DNA.